Hey guys, welcome to our basic genetics video. We're just going to be teaching you some things about genetics and we'll do a little lab. So first we're going to start with Gregor Mendel. He was the father of genetics, and he was called the father of genetics because he created these 3 laws that people are still using today. So first we're going to start with his first law he created, which was the law of dominance and recessiveness. So this basically means that some alleles which control different traits, like the dominant alleles control how a trait is outcome on to a physical characteristic of your body. So it shows up in organisms, and it covers up the other alleles, like the recessive alleles. And
So the next law is the principle of segregation, which states that 2 alleles for each trait separate when the sex cells are being formed. So if you have an organism who has the alleles big T, little t, the sex cells that it forms will have, one will have big T, the second will have big T, and then the last 2 will have little t's. And then so this big T, little t shows a heterozygous trait, which means that it has a dominant and a recessive, which is where the law of dominance and recessiveness comes from.
Okay, the last law that Mendel created was the principle of independent assignment. And this means that the outcome of one characteristic does not affect the outcome of another. So right here, Emily wrote down the trait for an ear, and then the trait for how tall you are. And the trait for the ear does not affect how tall you become. If you have a homozygous recessive, which is what this ear is, allele, then you won't become like heterozygous tall. Okay, going back to what we said earlier about the heterozygous tallness trait. We have heterozygous alleles, homozygous dominant alleles, and homozygous recessive. So heterozygous means that you have a dominant and a recessive allele. The dominant allele covers up the recessive allele, so it still shows up in your characteristics as a dominant trait. The homozygous dominant allele is 2 big T's. So it's going to be both dominant alleles from the parents. And then a homozygous recessive is 2 little t's, which means a recessive allele from both parents.
Cool.
Okay, so the last thing we're going to talk about is codominance and incomplete dominance, and this was not discovered by Mendel. So we have a blue parent and a red parent, and they mate to create an offspring, which is the child that has a red and blue showing in their phenotype. And phenotype means the physical characteristics shown in an organism. So they have red and blue, so you see both, and this is codominance because both of them are dominant to show that they're red and blue. And then next is incomplete dominance. So now I have M&Ms here to show you what it means. So we have a red parent and we have a yellow parent, and they mate to make an orange parent. And that means incomplete dominance because they mix together to create orange, so not either one is dominant enough to show over the red or yellow.
So the next thing we're going to talk about is a monohybrid cross. Now, monohybrid cross shows what genotype, which is what alleles an organism will have, based on what its parents' genotype was, which is their alleles. So, if you take a heterozygous parent, if you take its alleles for tallness, which would be big T, little t, and you cross it with a homozygous recessive parent, it will show you what their offspring will look like. So in this one, it gives you heterozygous, homozygous recessive, and homozygous recessive. And the heterozygous ones, keep in mind, show the dominant trait for those alleles. Now that we've explained all these simple genetic things, we will now do a short lab going over everything we've learned. So for this lab, you will be looking at mouse fur color alleles. So the blue M&Ms will represent dominant alleles, and the green M&Ms will represent recessive alleles. So before you start, you're going to want to put 5 blue M&Ms and 5 green M&Ms into a cup. For the second half of the lab, you will be looking at flower color. and incomplete dominance in flower color. So you're going to want to put 5 red M&Ms and 5 yellow M&Ms into a different cup.
Okay, so for part 1, we're only going to be dealing with the blue and green M&Ms. So we're going to pick 2 random M&Ms, and for me, I got a green M&M and a blue M&M. And the blue one, remember, is dominant, and the green is recessive.
So those 2 alleles represent the first parent. So then you pick a second set of M&Ms, and I also got a blue M&M and a green M&M, and that represents the second parent. So because the blue M&M is dominant, it represents a big B for black fur color in mice. And because the green M&M is recessive, it represents a little b, which would be white fur color. And so if we take our first parent, which is blue and green, which translates to big B, little b, that is the first parent's genotype because it is the 2 alleles that make up a trait of the organism. The second parent, who has the same genotype, which is big B, also little b, this would be the second parent's genotype for that particular trait.
Okay, now write down what you got, like Emily did here.
For the
so
parents.
if you had a green and green, that's 2 recessive, so it'll be little b, little b. If you had blue, blue, it'll be big B, and you had heterozygous right here, big B, little b. So just write whatever you get for both parents, and then we're going to cross.
So crossing on a monohybrid cross is when you bring down the alleles next to each box into their row or column that they're associated with. So you'll bring this B down, and you'll bring this B over, and then you'll bring the B all the way over, and this B correlates to this one, and as you see, so a little b will be here because on each side of the rows and columns there's a little b and a little b, and it all correlates to itself.
So this right here is the possible genotypes of the offspring of from the parents. So we have big B, and we have big B, little b,
Which comes from both of these.
and then we have little b, and we have 1 out of 4 for big B, 2 out of 4, which equals 1/2, and then 1 out of 4. And this is the possible genotypes that the offspring could have.
So if you translate these into percentages, it shows you the percent of possibility that there is that this parent will have, let's say if you cross 2 heterozygous mice, the possibility that they will have a white mouse, which is little b, is 1 out of 4, which is 25%. So there's a 25% chance.
This is the possible genotypes, but then we can do the possible phenotypes, which is the physical characteristics. Since dominant alleles cover up over recessive alleles, the heterozygous alleles will show out to be black.
Yes. Okay. So the heterozygous alleles and the homozygous dominant alleles will both show the black phenotype.
So we have 3 out of 4 that could be black.
So 3 out of 4 of them are black, which would be these 3,
And then
which is 75%. But then only 1/4 of them will be white, which is 25%, and that's this one down here. So you're going to take the cross based on the parents that you picked from the cup and the percentages of the genotypes and phenotypes, and you're going to write that onto your lab paper, and then we'll move on to the next part of the lab.
So now we're going to pick a random offspring. To do this, pick a number 1 through 4. This is going to be 1, this is going to be 2, this is going to be 3, and this is going to be 4. Whatever number you picked will be whatever genotype that you got from your cross. For us, we got number 2, and this has big B, so this is the one we're going to be using to move on. So this is the genotype we got from our random selection. And
the next genotype will be chosen from the cup that you had originally, and you can put the M&Ms from the last drawing back in so that there's an even number. And then you're just going to pick 2 random ones. I got 3. And so I got 2 greens, which, because greens are recessive, that means that our second parent for this crossing will be little b. And then, so she's going to draw the cross real quick, and you're going to cross those. And so this is This is from your F1 generation, and then the offspring of these will be from your F2 generation. And then you'll have your lab, and you'll just copy all this down just like you did for the last one.
Okay, so like we did before, it's gonna be big B, little b goes to this square. Big B, little b. And that's the same exact thing because we have two big B's and two little b's.
They're both homozygous, and when you cross them, it gives you all heterozygous.
This is homozygous dominant, and this is homozygous recessive.
So now we're going to do the same thing that we did last time after you've written down all your phenotype and genotype percentages, and you're going to pick a random number 1 through 4. But this time, after you've picked your number, we're going to go, we're going to go 1, 2, 3, 4. So now it's different. We changed it on you. And that's going to be the parent of your third generation, and you will cross that parent with a random parent from your cup again. And then you'll have 3 generations of offspring showing different colors. So for the second part of the lab, we're going to have a cup just like we did the first time, but it's going to have a red and yellow M&Ms in it. So you're going to do the same thing like you did earlier, where you're going to pick 2 random M&Ms. And so my first 2 are red and yellow. But this part of the lab is on incomplete dominance in flowers. So if a flower has a red allele and a yellow allele, because the dominance is incomplete, those colors are going to mix. To give a visual representation of a yellow allele and a red allele in a flower, you can take an orange allele, which shows how the colors mix, and instead of being covered up by one
Because it's
allele,
incomplete dominance.
Because it's incomplete dominance, rather than being covered up by one allele or being spotted, which is what would happen in cocoa. So the genotype for our first parent is R and Y. And keep in mind that even though the phenotype is orange, the genotype is still the alleles that you would get. So for the second parent, we're going to do the same thing that we did, and we're going to pick, wrong cup, we're going to pick 2 M&Ms. Here you go. Pick 2 M&Ms. And so our second parent for this cross is 2 yellows. So we're just going to write that, and then I'm just going to draw our cross real quick, and then we'll be able to cross them and show what an orange flower
mixed with a yellow
and
flower.
a yellow flower.
Showing incomplete dominance.
Correct.
Because R and Y is an orange flower, just so you know.
Yeah. So the first thing we're going to do is it's just like the last cross we did. You pull down the R in both things, and then you pull over the Y's, and you pull down this Y. So there are the genotypes for this set of parents. So the genotype for RY is half of them, which is these 2, and the genotype percentage for YY is also half, because it's these 2, which is 50% each. And then the phenotypes, you'll see we have no red flowers in this crossing because this parent has no red alleles in it. The phenotypes for this will be yellow for the 2 double Ys, and orange for the red and the yellow, the R and the Y, and the second R and the Y. So 50% are orange and 50% are yellow.
Okay, so now you're going to pick a random number 1 through 4 again for the offspring. So
you picked your number, and then we're going to go 1, 2, 3, 4. And then whatever number you picked, there's your parent for the next generation. So the next steps you're going to take are very similar to the green and the blue M&Ms, where you would take your randomly picked one from here, and then you'll take your cup, and you'll pick 2 M&Ms from your cup, and you'll cross those, and then you'll take another random one from that crossing.
And you write down your data for the phenotype and genotype.
On your worksheet, you'll have places to write down all of these things that you have and all the data that you found, so that you can answer the questions that are at the end of the lab.
Thank you so much for joining us today in our genetics lesson.